Good evening. My name is Anita Pepper, and I'm Vice President of Institutional Advancement at the Wistar Institute. It's truly a pleasure to gather virtually this evening, much to our dismay, as I get the honor of introducing you all to Dr. Akiko Iwasaki, the Waldemar von Zedwitz Professor of Immunobiology and Molecular, Cellular, and Developmental Biology, Professor of Epidemiology, Professor of Molecular, Cellular, and Developmental Biology at Yale School of Medicine, and investigator of the Howard Hughes Medical Institute. And we get tonight to present her with the 2021 Wistar Institute Helen Dean King Award. I want to start this evening by giving my sincere thanks to the Women in Science Committee, who's a wonderful group that have been critical to this program, and to our sponsors, including our lead sponsors, Glenn Mead, Pfizer, and Constance and Sankey Williams, and all of you who have generously supported this initiative in Wistar Science. Wistar is an international leader in biomedical research and training, home of breakthrough science discoveries in cancer, immunology, and infectious disease research. Wistar's mission as an independent research institute is to let our scientists focus solely on the science. We give our researchers financial support and a collaborative environment to make new discoveries in biomedical science and to push those discoveries from the bench as therapies and solutions for people and improve human health. Wistar's Women in Science program highlights the crucial role that basic and clinical research plays in addressing the scientific challenges related to health and showcases the distinguished and dynamic women behind the science. When biomedical research was an entirely male-dominated field, Wistar hired the first female scientist, tonight's namesake, Dr. Helen Dean King. She started working at Wistar in 1908 and at the time was the only woman in America to hold a full professorship in research. She spent the entirety of her career here at Wistar and until her retirement in 1950, where she made seminal contributions to science, such as the development of the laboratory model still used and known today worldwide as the Wistar rat. Named in her memory, this award is presented annually to a transformative scientific pioneer working in the field of biomedical research. Like Dr. Helen Dean King, Dr. Iwasaki is a forward-thinking, boundary-pushing scientist, and I'm thrilled to present her with the award tonight. Dr. Iwasaki received her PhD in immunology from the University of Toronto and completed her postdoctoral training with the National Institutes of Health before joining Yale's faculty in 2000. She's received numerous awards and honors, and I'm only going to mention a few, which include the Burroughs Wellcome Fund Career Award in Biomedical Sciences, the Wyeth Young Investigator Award, the BD Bioscience Investigator Award, and the Seymour and Vivian Milstein Award for Excellence in Interferon and Cytokine Research. Dr. Iwasaki has been an investigator of the prestigious Howard Hughes Medical Institute since 2014 and was elected to the National Academy of Sciences in 2018 and to the National Academy of Medicine in 2019. Dr. Iwasaki has spent her career learning about how the human immune system responds to viral infection. She is currently directing a translational immunology team to investigate the role of the immune response in COVID-19 disease outcomes, in addition to a wide breadth of work working on herpes viruses, influenza, even Lyme disease, among others. Her team recently published a study on the development of an RNA-based therapeutic that could lead to the treatment of immunocompromised patients and offer additional protections against SARS-CoV-2. In her spare time, Dr. Iwasaki is a prolific contributor to public education on immunology and infection, acting as a sought-after voice of information for her expertise on viral infection, especially COVID-19, with op-eds published in outlets such as the New York Times. She's also well known for her advocacy of women and underrepresented minorities in STEM, using social media as a tool to draw critically needed attention to these important issues. I highly recommend following her on Twitter. It is with great pride that we now present and congratulate Dr. Akiko Iwasaki with the Wistar Institute Helen Dean King Award, honoring outstanding women in science for their courage, leadership, and contributions to advance human health. Akiko, I give you the Wistar rat.
Wow, thank you.
And now I would like to turn the screen over to the 2021 Helen Dean King Award winner, Dr. Akiko Iwasaki, to hear about immune responses to SARS-CoV-2.
Thank you so much. Thank you so much, Anita, for the kind introduction and the wonderful rat sculpture here. And I'm going to show my screen. All right, you can see my slide, right? Okay.
Yes.
Okay, wonderful. So yes, I'm really honored today to be giving this Helen Dean King Award Lecture 2021. And it's really people and women that came before us, like Helen Dean King, who really led the way and paved the way for the rest of the women to be able to enter science and succeed. And I really wish to do the same for the next generation of scientists. So it's such an honor for me. And I thank you for this honor tonight. So I'm going to talk about immune responses to SARS-CoV-2 and what we've been learning over the last almost 2 years about this virus and the infection and immunity. So we've been studying, along with many, many other scientists, about the COVID-19 disease. And one of the striking things about this infection is that it really has a huge heterogeneous outcome depending on the person that's infected. And it's not necessarily that the age and sex determines all the outcomes. Even amongst the same age group or the same sex group, we're seeing very different outcomes with the infection with SARS-CoV-2. People either get asymptomatic infection or symptomatic infection that go from mild to fatal diseases. We also have infections that are acute, where people recover completely from the infection. And there are long haul infections, people who even start with milder disease, but turn into a very long haul debilitating disease symptoms. So some of that has to do with, as I mentioned, age and sex. However, it's not the only answer. For instance, if you look at the moderate, severe to fatal disease groups, we've identified different factors. For example, even the timing of the neutralizing antibody shift the ability of a person to recover from infection or have fatal disease. We've also demonstrated that saliva is a much better prognosis for saliva viral load is a much better predictor of a disease outcome than, say, a nasopharyngeal swab viral load. We've also demonstrated that the central nervous system could become target of infection using the mouse model, human organoid, and patient samples. On the other hand, we've also focused on some of the key differences in immune responses that occur in female and male patients, demonstrating that early immune responses in the female patients are characterized by activation of T cells, whereas male patients, especially the older male patients, really fail to activate T cells early on, and that results in the worsening of the disease outcome. We've also been studying the impact of COVID-19 during pregnancy and shown that in one case, it resulted in a severe impact on the pregnancy, having the virus infect the cells of the placenta. And unfortunately, that pregnancy, there was a fetal loss in that pregnancy. And in other pregnancies in which COVID-19 remains mostly respiratory, we've also seen differences in the placenta with respect to different transcriptomes that are expressed in that tissue distal to the site of infection, potentially indicating impact in pregnancy and development of the fetus while the COVID-19 disease is occurring within the mother. So today what I'd like to do is to focus on 4 separate stories, all related to COVID-19 and immunity. In the first part, I'd like to describe the basics of how the immune responses change over time during COVID-19 in hospitalized patients. Then I'd like to go into vaccine-mediated immunity and how it confers protection across different variants of concern. And then I'd like to get into the mouse model that we've developed to dissect the different types of adaptive immune cells and effector functions that control SARS-CoV-2. And finally, I'd like to talk about an ongoing work on long COVID and possible disease pathogenesis. So this human and translational immunology investigation all started back in February of 2020 when we knew that the virus was coming. At that time, we didn't have a single patient in the hospital, but we knew that the virus was spreading. And so we formed a biorepository called Impact Yale, where we can consent and enroll patients or healthcare workers to collect their biospecimen over time. So the inpatient cohort, we were collecting every 3 days, blood, saliva, and B-swab, feces and urine and other samples. In the second patient cohort, the outpatient cohort, we also collected biospecimens over time. And then the healthcare workers served as a negative control, healthy control, but some of them became exposed and infected during the course of investigation. And we were the first to be able to alert to them about their status because at that time, there was very limited testing available. So using this Impact Yale cohort, we first analyzed the immune responses that are happening in the patients during their disease course. And these four brilliant trainees, John Klein, Patrick Wong, Carolina Lucas, and Tiago Castro, led the study as co-first authors. The study design was to collect biospecimen and analyze immune responses and viral load over time in people who were hospitalized with COVID-19. And we divided the patients into different clinical scores, 0 being the healthy control, 1, 2, and 3 being people who are hospitalized in the non-ICU settings, who required nevertheless supplemental oxygen, 2 and 3. And then 4, 5, and 6 indicate patients who were in the ICU who received supplemental oxygen, 5 being the mechanical ventilators, and 6 being death. And we monitored immune responses over time. One of the first very clear evidence of what was happening to these patients was the significant T-cell lymphopenia that we observed. So in all these graphs, the healthcare workers are in green, and then the navy indicates the moderate patients, and the pink is the severe patients. And you see that T-cell numbers significantly drop in patients who are hospitalized with COVID. And inversely, we start seeing monocytes, especially inflammatory monocytes, come up in their peripheral blood, and also low-density granulocytes such as neutrophils and eosinophils that come up within the PBMC fraction. We also detected a number of pro-inflammatory cytokines, IL-1, IL-6, TNF, interferon, and so on. And here we also see antiviral factors such as interferon alpha being elevated as a result of COVID-19. We also see chemokines and other cytokines as well. So in looking at all these factors, what we realized is that the viral load in the nasopharynx is very different between severe and moderate patients. In the severe patients, the level of viral load, as you can see in the pink bar here, is maintained at high levels over the course of disease. Whereas patients with moderate disease, they were able to control the viral load over time, and some of these patients clear the virus. And when we look at the antiviral interferons, such as interferon alpha and gamma, what we see is that instead of the interferon controlling the viral load, it appears that the viral load is driving the interferon, as there is a positive correlation between viral load on the x-axis and the level of these interferons on the y-axis. And similarly, pro-inflammatory cytokines such as TNF and TRAIL are also increasing as a result of viral titer. When we expand the analysis to other cell types and factors, what we find is that the type 1 immune responses, which is typically found in viral infections, are elevating in the severe COVID patients. However, we also started to see type 2 immune responses. These are the immune responses we typically think of for parasites and allergy. That's also becoming elevated. Eosinophils, lots of cytokines associated with type 2 immunity, as well as IgE, certainly elevated in the severe COVID and not in the moderate COVID. And we've also seen the type 3 immune responses that are dedicated for fungal infection and extracellular bacteria also elevating in the severe COVID. So this is a slide I usually use every year to teach immunology to medical students at Yale. And I tell them how specific the immune response is by generating specific types of T cell subset based on the kind of pathogens that the antigen presenting cell recognizes. And these antigen presenting dendritic cells secrete first order cytokines that inform the naive T cells to become certain kind of T cells that are specific for or particularly targeted for the parasite response, Th2, Th1 being viral, and Th17 being antifungal. However, in the severe COVID patients, all of these cell types are engaged at the same time. And this is a very unusual outcome of a virus infection. We then went on to look at the different types of plasma factors that are found in the patients. So each patient is indicated by a column, and each row represents different factors that are found in the plasma. And what we did was to do an unsupervised clustering analysis of the patients based on the plasma factors, and found that there are 3 clusters of patients. First cluster of patients in the middle have the highest levels of plasma factors belonging to growth factors and tissue repair factors. Here is at PDGF, EGF, VEGF, IL-7, and so on. So the darker the color, the higher the level of these factors. Second cluster of patients, in addition, had elevated levels of chemokines and mixed cytokines. And third cluster of patients had the highest levels of type 2 and 3, as well as mixed cytokines. And when we followed these patients over time, what became clear is that the first cluster of patients were able to control disease clinical score over time and become discharged, whereas the second and third clusters of patients developed worse and worse disease outcome, and they had the highest number of coagulopathy and mortality. And when we look for all the factors we've measured and asked for biomarkers for COVID-19 mortality, we found IL-18 to be the top biomarker on the very bottom here. IL-18 is a cytokine released as a result of inflammasome activation. And so this suggested that there is inflammasome activation that's occurring in COVID patients that are predictive of mortality. Interferon alpha was the number 2 biomarker for mortality. And this is quite ironic because interferon alpha is the very cytokine that we need to fight off the infection. And what it indicates is that this virus is quite good at evading the antiviral effect of interferon. And instead, the interferon is driving some of the disease pathogenesis by recruiting innate immune cells and inducing more inflammatory responses. So this study, we've learned that persistent viral load is detected from severe disease patients, and it appears to be the viral load that drives the interferon and cytokine responses, and that this increased levels of interferons and cytokines are associated with mortality. And the plasma factors alone can distinguish patients into those who recover versus who go on to develop worse disease symptoms. And the features associated with recovery was consistent with the tissue repair type of program. And finally, in patients who developed worse disease had a mixed cytokine responses. And this really reflects a maladaptive immune responses that occur that may be driving the immunopathology in these patients. So these patients are also are likely to have failed to control the viral load earlier, and that these maladaptive immune responses are a reflection of the compensatory mechanisms that are employed to try to control the virus and yet resulting in pathology instead. So we've learned a lot about these immune responses happening in the patients, but what about vaccine-induced immunity? And in particular, the question what we wanted to address was, how do variants of SARS-CoV-2 affect the vaccine-induced immunity? And this work, again, was led by Dr. Carolina Lucas, along with two other first authors, Chantal Vogels, who is a postdoctoral fellow in Nathan Grubal's laboratory, who does a viral surveillance in the community, and then Dr. Insi Elderam, who is a pediatric infectious disease doctor, who recruited the participants and designed the study along with us. So the study design is to recruit healthcare workers who either had prior infection in blue or who had not been infected in purple. And we collected their blood samples before and after 2 doses of mRNA vaccines, both Pfizer and Moderna. At 7 and 28 days post first dose, 7, 28 days or 70 days post second dose. And we compare the immune responses against the SARS-CoV-2. What we found is that the mRNA vaccines, especially after 2 doses, are able to elicit very robust antibody responses in all these recipients. So on the top panel is the SARS spike-specific IgG, and the second middle panel is the S1 region of the spike, and then the bottom is the receptor binding domain of the spike. These are the antibodies against these regions. And we divided the patients or participants into those who were previously infected in blue and those previously uninfected. And when you look at the blue participants, there are some baseline antibody levels in these patients, participants. However, this level goes very high after the first dose of mRNA vaccine, and it's maintained at very high levels throughout. The participants who were never previously infected are also able to generate very good levels of antibody, especially after the second dose. We also measured neutralizing antibodies to the ancestral SARS-CoV-2 using authentic neutralization assays using live virus. And here what we find is that, again, some of the participants who had been previously infected had some levels of neutralizing antibodies to begin with. However, this level goes up quite dramatically after the first dose of mRNA vaccine. And then the ones that never had the infection are able to catch up after the second dose of vaccine. We also looked at T cell responses, both the CD4 T cells as well as the CD8 T cell responses, and found that T cells are indeed well activated against the spike, both the original lineage A spike as well as spike of a P1 variant of concern, which originated in Brazil. And you can see that the red square indicates the T cells that are activated, and that the spike proteins of both types are able to activate T cells upon vaccination. However, when we look in the CD8 T cells, the previously infected people were able to generate very good CD8 T cell immunity against the ancestral spike. However, even the people who weren't infected after vaccination are able to elicit good CD8 response. But the P1 specific CD8 T cell responses were reduced as a result of the mutations that accumulate in this spike. So the ancestral spike, the cross-reactivity is reduced for the P1 variant. We then wanted to understand the neutralization capacity of the antibody generated by the vaccines against all the different variants that we find. So many of these isolates were actually collected from the surveillance program that the Grubal lab has. And Carolina purified and amplified these variants to be able to test them against the neutralization. And also indicated in the in this panel A is the key amino acid differences that occur within the spike protein of the different lineages of variants. And on the right of that panel is the PRNT50, the neutralization titer in log scale. And what we find is that, of course, the vaccine induced the best neutralization titer against the ancestral lineage, which is the lineage that the spike antigen comes from for the vaccine. But the neutralization was maintained pretty high level against the Iota and the Alpha variants. It's only when we get down to the Delta variant that there's a significant reduction in the level of neutralization elicited by the antibody, by the vaccines. And this coincided with mutations such as the L452R mutation. And in fact, on the panel B, what we're showing is the estimated effect of different amino acids that are found in the spike protein and their ability to escape neutralization. And the L452R that is found in the Delta variant is the single most impactful mutations to escape neutralization. And you go down this list even further, and what we find is that there is even a more significant drop in neutralization of the gamma and the beta variant. And this coincides with mutations such as the N501Y combined with the E484K mutation, as well as some of the N-terminal domain deletions. However, when we look at the inter-individual variations of neutralization antibody, there is a much bigger variation between individuals than between the variants with respect to neutralization. So even though these people are fully vaccinated 28 days after the second shot, some people developed no detectable neutralizing antibody, the two people down here, while others developed very high levels of neutralization. And those who develop very high levels or tend to remain a very high level across different variants. Those who develop very low levels tend to drop off in their activity for neutralization as variants accumulate more and more mutations. And we can divide these participants into those who are previously uninfected on the top and those who are previously infected on the bottom. What you see is that there is a significant advantage of having had the infection prior to vaccination. That is because the infection itself is serving as one dose of antigen. So on the bottom, people really had 3 encounters with the spike protein. And what you see is that not only are the levels higher to begin with, but they're able to resist the different mutations that accumulate in the variants until you get to the gamma and beta variants. Whereas the previously uninfected but fully vaccinated people generate a good response against the ancestral spike. However, this leads to a much quicker drop-off in the neutralization titer, especially when the Delta variants emerged. So this study showed us that the mRNA vaccines are indeed pretty amazing at inducing robust antibody and neutralizing antibody, as well as T-cell immunity. And that the prior infection does confer advantage to mounting a higher levels of these antibodies and T cells, and that are still cross-reactive to variants of concern. And distinct mutations do confer different degree of escape from neutralization. And there are also certain combinations of mutations that synergize or antagonize against neutralization. And finally, we believe that the viral genome surveillance efforts are important, not only to understand which variants are circulating, but which variants may be circulating with breakthrough potential. Now that we have all these amino acids analyzed, we can predict what kind of mutations might confer this resistance against the neutralization. And when we do, we can design a variant-specific booster vaccine. So I focused a lot on the neutralization, but indeed there are other mechanisms by which adaptive immune response can confer protection against the virus. So a infectious disease fellow, Dr. Ben Israelo in the lab, asked this question: what are the determinants of protective immunity against SARS-CoV-2, using a mouse model that he developed in the lab. What he did is first to ask the role of B and T cells in clearance of the primary infection. And to do this, he employed a mouse model in which he can transduce mouse of any genetic background with human ACE2 using the AAV vector. And this enables any mouse line to become infected with SARS-CoV-2. And then he can analyze the viral load and immune responses over time. So he took wild-type animal or mu-MT mice, in which there are no B cells present in these animals, and transduced both of these with human ACE2, and then infected later on with SARS-CoV-2 and analyzed infectious viral particle in the lung. What he found is that in the wild-type animal, all the mice were able to control the viral load completely within 7 days. In the mu-MT animals, all the animals did control the viral load within 14 days. In 7 days, half the animals still had infectious viral particles. This indicated that B cells are not necessary to control primary infection. However, when Ben repeated this experiment in the RAG knockout mice, which are devoid of B and T cells, then these animals are no longer able to control the infection at all, and they become chronic carriers. And this illustrated that adaptive immunity is indeed necessary to control primary infection. He then turned his attention to T cells and asked, what is the role of T cells? By depleting the T cell subsets CD4 and CD8 or both prior to challenge with SARS-CoV-2. He used first wild-type animal to ask this question, and what you see is that infectious viruses are completely absent from animals that are depleted of CD8s or CD4s, indicating that when you have B cells around, depletion of CD4s and CD8s have no impact on the clearance of the virus. When he depleted both CD8s and CD4, he started to see some of the animals having infectious titers. And this is in part due to the fact that these T cells are necessary to support the B cells in secreting antibody against the spike protein. And when he did the same set of experiments using the mu-MT mice that are devoid of B cells, he then noticed that the CD8 depleted animals, so these red bar indicates animals that only have CD4 T cells to fight off the infection. In that setting, he did see some animals actually are unable to clear the infection. Similarly, in the mu-MT mice that are depleted of CD4 T cells, in which only the CD8 T cells are available to fight off the infection, again, there were some animals that still remained some viral infection. However, when he depleted both CD8s and CD4 from the mu-MT mice, now that these animals have no adaptive immunity, and just like the RAG knockout mice, none of them could clear the virus. So what about the role of B and T cells in protection against reinfection or vaccine-mediated protection? To address the reinfection issue, Ben first took an approach of an adaptive transfer model in which he collected convalescent serum or convalescent T cells from animals that have recovered from a prior infection with SARS-CoV-2. Serum transfer to RAG knockout mice, these are devoid of adaptive immune cells, enabled him to look at the sole effect of the serum antibody in conferring protection. And indeed, these animals that received a convalescent serum were completely protected from infectious virus within 7 days. In T cell transferred animals, some mice did completely clear the virus while others did not. Even the level of the viral load, even though the level of the viral load was much lower, indicating that T cells are partially effective, whereas antibodies are completely effective in protection against infection. Ben then turned to a mRNA vaccine model to create a setting in which animals had different load or different titers of antibodies against the spike protein. He created this cohort of mice by immunizing mice with different doses of mRNA vaccine, all the way from 1 microgram down to 0.01 microgram. You can see there's a beautiful dose-dependent response of antibodies generated, 1 microgram being the best, followed by 0.3, 0.1, 0.03, and by the time he got down to 0.01 microgram, there was no detectable antibodies. He took these animals and challenged them with a lethal dose of SARS-CoV-2. And what he found was that in animals that received 1 or 0.3 microgram of vaccines, they were completely protected from disease or death, whereas animals that received 0.1 in green, more than half the animals succumbed to infection. And the ones that received 0.03 or 0.01 were not at all protected. So knowing the amount of antibodies that were generated prior to challenge, Ben was able to calculate the correlative protection based on antibody levels. And there's a very nice linear relationship between the amount of antibody generated and the prevention of weight loss and survival. So this gave us a good correlative protection based on spike-specific IgG as well as neutralizing antibodies. Finally, Ben wanted to understand the importance of CD8 T cells in protection against reinfection or mRNA vaccine-mediated protection. He created 2 cohorts of animals, one that are fully vaccinated with 2 doses of mRNA vaccine, and one that had been infected and recovered, convalescent mice. And then we challenged these animals with SARS-CoV-2, either ancestral lineage or the beta variant. And half the animals were depleted with anti-CD8 antibody, and other half remained intact. And all of these animals elicited very good levels of antibodies. And when challenged with lethal dose of these viruses, all the animals that were vaccinated or convalescent were protected, regardless of whether they were depleted of CD8 T cells or not. And in fact, when he looked for infectious viral particle in the lung of these animals, all vaccinated animals and all convalescent animals were completely devoid of infectious viral particle, whether they were challenged with the ancestral lineage or the beta variant. So this study illustrated the relative importance and the compensatory roles of T and B cells in clearing primary infection, that in the absence of one, the other can compensate. However, in the absence of both T and B cells, animals are unable to control primary infection and remain chronically infected. Adaptive transfer of the immune sera or effector T cells can provide protection, either complete or partial. And that the convalescent mice or in immunized mice, CD8 depletion had very little impact on protection against challenge by SARS-CoV-2. And finally, we were able to calculate the antibody levels required for protection induced by mRNA vaccines. So what this illustrates, along with so many other studies on vaccines and immunity, is that even though mRNA vaccines were not designed to elicit mucosal immunity, there is sufficient crossover to provide a very good level of protection against infection and replication. So what we're now learning is that the mucosal surfaces of mRNA vaccinated people contain some levels of antibody, IgG or IgA, against the spike in order for that to block infection. And replication and spread can further be blocked by T and B cells. And we also know that symptomatic infection is dramatically reduced by vaccination. And that would also reduce the transmission activity, such as sneezing and coughing. And finally, in immune individuals, the emitted virus may be also not as infectious if they were to be coated by antibodies. So the mRNA vaccines are turned out to be very good in not only reducing severe disease, but infection, even though the Delta variants are challenging this view because of its highly transmissible nature. So what we need in the future is to develop mucosal vaccines that would prevent infection altogether, and so that we also prevent onward transmission from the infection. Finally, in the last few minutes, I'd like to talk about our ongoing work on long-term consequences of COVID. And you probably all heard about long COVID or know someone with long COVID. It's a pretty common outcome of a COVID-19 infection. So people with severe COVID who were hospitalized and discharged, at least 50% or more of these patients are currently suffering from long-term symptoms after months. And this is also typical of what happens in post-ICU syndromes, where regardless of the reason for the ICU admission, many people suffer a lot of neurological symptoms. However, it's also becoming clear that even mild or asymptomatic COVID can lead to long-term symptoms. And the percentage differ between different studies that you read, anywhere between 5 to 30%. And these kinds of post-COVID-19 syndrome is occurring much more so in women of middle age, whereas the severe COVID post-syndrome is occurring in older males, because those are the ones that are also susceptible to severe COVID. There are over 200 symptoms reported for COVID-19 and long COVID, including organ systems that involve systemic, neuropsychiatric, head, eye, ears, and nose and throat, as well as endocrine, cardiovascular, pulmonary, musculoskeletal, and GI tract. And many patients are reporting over a dozen symptoms that are indicated here, while others have 2 or 3. So it really depends on the person, and there's a spectrum of disease severity. When you follow the different spectrum of disease severity, what you see is that in the first few weeks of COVID-19, post-COVID-19 syndrome, the very severe and the severe numbers decline, which is replaced by mild and moderate and very mild symptoms. But you see that, unfortunately, these moderate symptoms and milder symptoms stay up and are not coming down over 7 months and beyond. As I mentioned, people suffer from different ranges of symptom. Those who start out with a little bit lower levels, number of symptoms, tend to recover within 90 days, whereas those who started with over a dozen symptoms tend to increase in their symptom number, and they don't recover within 90 days. We have a couple of hypotheses about long COVID disease. We think that it's possible that there are viral reservoirs that are established in the body that is not in the nasal cavity or the oral cavity, because they're all PCR negative. But it's possible that the live infectious virus or viral remnants may be resident in different tissues that's triggering chronic inflammation. Another possibility is autoreactive lymphocytes that's being activated after an acute viral infection. And depending on the specificity of these receptors, they can target different tissues and have different impact on pathogenesis. There's also other hypotheses like tissue damage that's incurred by the original infection, dysbiosis of microbiome, and latent viral reactivation, particularly the herpes virus family members. We do see evidence for viral reservoir that is seen in COVID patients after weeks or months of having had the infection. This is a biopsy from intestine from a COVID patient 92 days from the symptom onset. And you see this beautiful green staining here in the ileum and duodenum of nucleocapsid from SARS-CoV-2. So 5 out of the 14 patients tested had positive staining. 3 out of the 14 had PCR positive for viral RNA after 3 months. Another study illustrates similar kind of presence of viral protein and RNA found in intestinal tissues as well as lymph nodes. So it's possible that these kinds of persistent viral RNA or antigen may be driving some of the pathologies. We have also observed many diverse array of autoantibodies during the acute COVID-19 disease. This was enabled by my collaborator, Aaron Ring, who developed a new technology called Rapid Extracellular Antigen Profiling, or REAP, where we are able to comprehensively analyze autoreactive antibody based on their yeast display library that expresses human exoproteome, which are proteins that are secreted or extracellular. And by binding those yeast cells with antibody and pulling down IgG positive fraction, and then you can decode the antigen using the barcode that's specific for these particular cells. Using this technology, we identified numerous targets, interferon being one of them, and many other cytokines and cell surface molecules. But we also identified a large number of autoantibodies that is expressed by different tissues. So each row here indicates different antigen. Each column is a patient. And the brighter the yellow, the more autoantibody there is in that patient. You see that there's a smattering of different autoantibodies against the CNS antigens, vascular antigens, connective tissue, cardiac, hepatic, and many other tissues. So indicating, and some patients actually have multiple autoantibodies across these tissues. So it's possible if these antibodies persist and are pathologic, that can explain some of what's going on in long COVID patients. There are a number of other reports in addition to what I just said, as well as in long COVID, there are some G protein coupled receptor specific autoantibodies that are being detected. So this is certainly something that we need to investigate further. So there is currently no treatments that are known to work to help the long COVID patients. However, there are a number of reports now illustrating that vaccines impact long COVID symptoms. So this is from a patient group called Long COVID SOS. They surveyed their patient base and found that about 60% of their patients are reporting improvement in symptoms after vaccination, regardless of the kind of vaccine that they received. Whereas about 18%, 20% are reporting worsening of symptoms. When you break these symptoms down into different categories, some vaccines appear to be better than others in reducing these symptoms, including the fever, Moderna being the best in reducing fever, muscle pain, and so on, while other types of symptoms are equally benefiting from vaccines. So how can vaccines end up improving long COVID symptoms? Well, one simple hypothesis is that the vaccine induces robust T and B cell responses that clear the viral reservoir, in which case you remove the source of inflammation. It's also possible that the cytokines and interferons that is induced by vaccination is impacting the autoreactive lymphocytes that then shut down their ability to provide toxic cytokines or antibodies. The latter type of response is expected to be temporary because you're not really removing the source of the problem, which is the autoreactive T cells and B cells. So currently, we have our mission to really try to understand immunological and physiological determinants of pathogenesis of long COVID. And we really hope to inform diagnosis and therapy. And as I explained, these hypotheses that we have about what's driving long COVID are not mutually exclusive. So some people may have collection of these things going on, while others are only driven by one or the other. So the idea is to really involve patients as equal partners in investigating these diseases. We have now established collaboration with Survivor Corps as well as patient-led, and also with different long COVID rehab centers, as well as new studies that we're recruiting patients into. And we're now doing flow cytometry analysis in real time to look at the cell composition in PBMC, as well as doing more deeper profiling, including the REAP that I discussed today, as well as cytokines and T cell receptor sequencing, and metabolomics and hormone profiling. And I am able to recruit brilliant minds to work with us on this. And then also, once we have the data set, we want to integrate these multimodal data set to be able to subset patients into different groups, potentially revealing the disease pathogenesis, so we can design a much informed and rational therapy for each patient. And ultimately, we want to return the data back to the patients so that they are also informed of their immune status. So I'd like to end here by thanking and acknowledging all the members of the lab as well as collaborators that contributed to the 4 different stories I was able to share with you today. And also I want to thank all my lab members who have been very inspiring to work with during the pandemic, as well as the Impact Yale cohort team, without whom none of the clinical work could have been done. And then many collaborators that we are working now together to solve the problem of long COVID. And this is a picture we took in early summer when all of us were fully vaccinated. And I'm going to end my talk here so that I can take some questions. Thank you.
Akiko, that was so amazing and impressive, and the wealth of information you just provided to all of us is just overwhelming. And I have my whole list of questions, but that's not my job. I'm supposed to relay everybody else's questions. So first, let me just say thank you for all that you and your collaborators have done, and all of the discovery science that has gone behind this incredible understanding of how this virus is infecting us and what we still need to learn. It's so profound. And we're really grateful that we have heroes like you working on this. So thank you so much.
Thank you so much. Thank you.
Really incredible. So one of the first questions that we have is, a lot of people have been asking, and this was not addressed as far as I know, but what are the genetic traits that may be behind some of these altered response and levels that you all are seeing kind of on the immunological response?
Yeah, that's an excellent question. That's not one of the focus of my laboratory, but people like John-Laurent Casanova and others are really looking into the genetics of disease and are finding sort of the key regulator of the innate immune response as being critical in preventing severe diseases. So if you're lacking or have mutation in genes that are involved in interferon responses, for example, you are at high risk for developing severe COVID. And so there is some evidence coming out in some of the extreme phenotypes, like the severe COVID. But going back to your question, like what determines this heterogeneity, it's likely not going to be one gene or maybe even one factor. It's likely a factor for about genetics as well as environment, diet, and also prior exposure to seasonal coronaviruses that may share some of the epitopes, and that may give someone advantage for developing better antibodies or T cell responses. So there's just a huge amount of complexity involved here.
Yeah, that's a wonderful answer. Thank you. So something that does pertain more to your work, one of the questions we've gotten is, what do you think about monoclonal antibody injections for prophylactic use in immunocompromised people?
I absolutely support that. I've been saying that from the beginning of the pandemic, as soon as the monoclonals became available. And I know that companies and people are now working on long-lasting monoclonals that can be up to 6-8 months of longevity. And those kinds of antibody can be injected, and people do have to get repeated injections, but not so often. And especially important for immunocompromised people who don't have any B cells or antibody responses. And there are many people like that, cancer patients, transplant patients, immunodeficient people, HIV. There are many instances in which people just can't mount the right kind of immune response. So the monoclonal antibodies, I think, are going to be great, but I guess you have to also do it in cocktail, because virus can evade very quickly if it's just one monoclonal.
Yeah, complicated again. So another question I had is, obviously, as your data has shown, the T cells and B cells are critically important to outcome. Can you explain to the audience a little bit about why we can't just get a test for our T cells and our B cells, and why this is just, I mean, everyone says, well, why don't we know about those? Why is it all about antibodies? Can you explain that a little bit?
Yeah, so I just did a Medscape seminar on this. It's a frustration, right? Like, yeah, exactly. Everyone wants to know, what is my level of T cells and antibody? And until recently, there really wasn't any kind of commercial test available. So we, as researchers, we can do all kinds of tests in the laboratory, but that's not, readily accessible to people. There are now a couple of T cell tests that's available that doctors can order, and they detect common T cell receptors that are stimulated in response to viral infection or vaccines. And so you can get a yes or no answer whether you have T cells or not. Obviously, you want more granular data, like, what kind of T cells do you have? How much T cells do I have? Is it the right cytokines that they secrete? That kind of granular information is still not available. But I think, if I was an innovator, I would make a company to test that.
Everyone out there, opportunity. That's great. Okay, so a lot of questions about boosters. And so one of the questions is long-term immunity for SARS-CoV-2 a possibility? And do you think that this third booster will achieve long-term immunity? And I know where you're going, but go for it.
Yeah, so I don't have a crystal ball, but what like, most vaccines are giving in more than one dose, right? And 3 doses is a typical vaccine strategy. So it's not unusual to need the 3rd dose. And indeed, this vaccine, mRNA vaccines and adenoviral, really benefit from the 3rd dose in boosting neutralizing antibodies and T cells and so on. So it makes immunological sense to get the 3rd dose. And especially if you're at high risk, this is really important now, because the Delta is, as I mentioned, highly transmissible, and there's a lot more breakthrough going on. However, whether we are ever going to need 4th and 5th dose and so on, that remains to be seen. The reason I say that is because the 3rd dose may be very good at maintaining high levels of neutralizing antibody against the current variants. But in the future, it's possible that a variant may arise where it escapes the existing immunity developed by the current vaccines. And that would require a booster that's specific to that variant. And so it's not really a booster in the sense that the same vaccine is given to you, but it's a booster that's required to elicit the right kind of antibodies and T cells. And that, I never want to say that's never going to happen, because we just don't know. We're still in the middle of the pandemic. So I would say that, we need to be humble, and we need to be ready to follow science when those things do arise.
Well, that is the perfect ending to a perfect night. So Akiko, thank you so much for all you do, for all of your work. Please thank your lab, and we're just grateful for everything. And congratulations on the Helen Dean King Award. And I wish everybody a good night. And I hope that we get to see you all in January when we have Diana Bautista joining us to talk about neurons, itch, and pain. So with that, it's, February 17th, 2022. I always think it's January. And with that, I want to wish you all a good night, and thank you for attending. So good night.
Thank you so much. Bye-bye.
Bye.